So good evening, everyone. Hope everyone is having a good day. So the title of my topic is From Molecules to Mechanism, Decoding Bovine Theileriosis through Integrated Omics. So before proceeding for the presentation, let me thank my lab mates, Dr. Paresh Sharma, he is the principal investigator, Akash Suresh, which is SRF, and Venkat JRF, who helped me to conduct this particular work. And I want to thank NIV for giving me this uh, facility to conduct my research, and as well as bioanalytics skill development forum for inviting me to give a lecture. So let me start with a small introduction to epicomplexion. The Theileria annulata parasite belong to this epicomplexion group of parasite, and these parasites are very unique as well as diverse, and these are also known as sporozoa, and they represent a vast group of unicellular protists with a broad environmental presence. These organisms are obligate intercellular parasite. That's why they need another host molecule to survive and replicate. And these parasites of very high importance for both human as well as livestock, as they cause very devastating diseases like Plasmodium cause malaria, Toxoplasma cause toxoplasmosis, and Theileria cause bovine Theileriosis in case of livestock. So this parasite belongs to this hematozoa, where one more parasite, that is Plasmodium, which is very important parasite causing malaria to human being. And Theileria is also belong to this particular hematozoa family. So now let's come to the bovine Theileriosis. So this TN infection is lead to bovine Theileriosis, where more than 200 million cattle are at risk of infection. This disease of a high economical importance, causing an estimated loss of $1,295 million per annum to Indian livestock. And the only drug that is available right now to cure this disease is bupropion. So some of the cancers hallmark associated with Theileria transformation are uncontrolled proliferation, resistance to cell death, as well as metastasis. So the research surrounding Theileria range from all aspects of biology. So this is a brief graphical representation of the central dogma, that is DNA followed by transcription, making of RNA, followed by translation, as well as protein. Okay, and but generally the cancer environment or tumor microenvironment is a product of misregulation or dysregulation at any of the following stages lead to cancerous phenotype. So there has been several studies that has done in case of Theileria to understand how this cancerous phenotype is working. So for example, start with DNA. People have shown recently that there is some DNA mutation associated with this particular transformation at the level of gene expression by conducting RNA-seq experiment, as well as individual proteins molecule has been studied to identify or dissect the cancerous phenotype. However, the current trend or current mechanism that is most important in regulating all the activity of protein is post-transcriptional modification, which has not been thoroughly studied in case of Theileria. So this is a screenshot I have taken from PubMed as well as HealthPress. So if you go by some keywords, phosphorylation plus cancer, you will find that almost 35,000 articles are there. And when it's come to epicomplexion, the number drastically reduced to 128. And when you come to Theileria, the number is 36 at the level of individual molecules. But if you look at a global perspective or to study the global phosphoproteomics, there is only one paper which only study the Saigon stage of the parasite and no study about the host molecules. That's why it's become very important to study this post-transcriptional modification to identify new molecules target to cure this particular disease. So in my study, we went for post-transcriptional modification, particularly phosphorylation to identify phosphorylation event at the global scale upon infection. So protein post-transcriptional modification play a very profound impact on all essential of cellular and biological processes. And the diverse PTMs and crosstalk is important for all sort of neoplastic transformation, carcinogenesis, as well as metastasis. So this graphic represent what are the output or what is the output of protein phosphorylation. So it changes the localization of the protein to change in the activity, turnover by making more or less, binding to different molecules for making a heterodimer so that it can become a transcription factor, uh, as well as PTM crosstalk between epigenetic modification as well as post-transplant modification. So protein phosphorylation is an important event which regulates all aspects of biology. So I'm moving to my objective slide here. So as there is a lack of study regarding the phosphoproteome of Theileria infection, so we basically asked this following question. Uh, so the first objective was to identify the total phosphoproteome of Theileria infected cells, that is for both host as well as parasite. Second is to assess the influence of parasite clearance on the phosphoproteome. So for example, what happens when you eliminate the parasite from the infected cells? Then the third question is, what are the kinases that are responsible for phosphorylating this the identified site from the proteomics data? Then what are the transcription factor or how the regulation of transcription factor are being done at the level of protein phosphorylation? Then we identified some key transcription factor and we try to validate by using wet lab. And we went for RNA-seq experiment to identify the target genes under the control of this particular transcription factors. So this is the study design. We have went for a non-nested design. So we have taken uninfected BL3 cells, infected TA cells, where you can see that the parasite is right now inside the cell, the red nucleus is there. And when you treat it with bupropion, which is a parasite-directed drug, there is an elimination of parasite and followed by cell death. And so we have taken three conditions: uninfected, infected, and BPQ treated. So we have taken two biological replicates in each condition and four technical replicates for each condition. So coming to the workflow, so we started with whole protein. So we have started with 2 mg to 5 mg of protein, followed by protein digestion by using trypsin. Then the peptides were the detergent removal was done by using C18 column. Now the C18 purified peptides were further taken for phosphopeptide enrichment. So we have used titanium dioxide enrichment kit from Thermo Fisher to enrich our phosphopeptide. Then this is the basic of mass spectrometry. So the four samples we have went for a 240 minutes run, and we have used this Q executive mass spectrometry to capture our data. So coming to the pipeline for identification and quantification of phosphopeptide. So after taking the raw fragments from the mass spectrometry, we use MaxQuant software to identify and annotate the protein or peptide, as well as to identify the variable modification, that is phosphorylation. So we have used both the databases, Bosch Taurus as well as Theileria annulata, to identify the proteome. Then the score calculation was done. This is a normal Andromeda score that you followed for uh, max quant. The maximum miscleavage we kept is 2. Minimum peptide length was 6. The fixed modification is carboamidomethyl. And we have taken variable modification phospho-STIY as we are more interested in this particular variable modification. Now coming to the statistical analysis. So we have followed a very stringent statistical analysis where we have only kept the phosphosite which have a high localization probability of more than 0.75 or equal to 0.75. Then we have done the intensity transformation by using log2x, and we have kept the valid values in at least one group. As phosphorylation can be a binary process, it can present or it can absent, or maybe or the expression may vary in a gradient. So we have kept this particular parameter at least in one group. Then we replace the missing value by using normal distribution or KNN. And for statistic or to identify ANOVA significant protein, you have used FDR 0.05 and randomization in 250. And the data normalization was done by using G-score. Now coming to the fast data, where right now the comparison is between uninfected cells and infected cells. So after we capturing and analyze our data, first we look for is whether our biological replicates are behaving in the same manner or not. So this particular scatter plot represent the intensity of different peptide that we have identified from one biological group. Here I have represented the control or infected one. the replicate 1 and replicate 2, and the Pearson correlation score is 0.83, which suggests that there is a high similarity between the biological group. Now coming to the identification of different phospho-STY site, so 86% of the identified phosphoproteome belong to serine, followed by T and Y. And this web logo represents the same thing that we have represented here by bar graph, that S phosphorylation is quite prevalent as compared to other two modifications. Now coming to the number of, so that's the second thing what we looked into our data is how many proteins are having more than one modification site or more than two or three. So 65% of the protein we identified are having one modification site, while 24% are having two modification. Around 8% of them are having three modification, and around 2% are having more than four. And when you look at the localization probability, the distribution is something like this. Around 934 phospho sites are having a localization score of 0.75 or more than 0.75. And for all our downstream analysis, we have only kept this particular localization score to identify the most stringent one. And this principal component analysis represent that the infected and the control ones are behaving separately. Now coming to the data analysis, or what the data we got from our whole phosphoproteome. So first thing what we looked into it is how many phosphosites are unique to infected samples, how many are common between the uninfected and infected one, and how many are unique to uninfected one. So majority of the protein, around 364 protein, which span across around 600 phosphosites, are unique to Theileria annulata infected samples. And when we performed a GOBP analysis of this particular unique site that present upon infection, what we found is there are many different pathways has been identified or differentially regulated, out of which I have highlighted three particular things, that is regulation of CAMK and AMPK signaling, positive regulation of telomere maintenance, as well as negative regulation of apoptotic process, which in turn says that this particular as this pathway are part of cancerous phenotype. So what we found from our data is our representation of this particular pathway and ultimately responsible for the cancerous phenotype. The next question we ask here is there any differential phosphorylation or expression changes of the phosphopeptide that we have identified from our data? So here we have only considered the class 1 phosphosite, which are having a localization score of more than 0.75. After keeping the multiple ANOVA test and FDA 0.05, we have identified 148 phosphosite that are differentially regulated upon infection. And one of the key protein that we identified from this differential phosphorylation data is phosphorylation of STAT3, which is a transcription factor at 705 position. And this 705 position has been associated with stability as well as changes in the localization of STAT3 molecules. And in order to verify that particular identification, we have performed an immunofluorescence assay to look for the localization. So this is an immunofluorescence data which suggests that the green one is the wild type STAT3, and this magenta one represent the phosphorylation STAT3. And you can see that DEPI here, this is the host nucleus, and this small dot represent the parasite inside the host environment. And we are finding a good localization of this particular protein inside the nucleus as there is a transcription. So whatever the phosphosite we have found from our data are quite stringent and have been verified using immunofluorescence assay. We have found out many different molecules from this data, which I'm going to discuss later in my slide. So now coming to the second aspect of our phosphoproteome study. So the initial study was comparing between three different conditions: uninfected, infected, and parasite clearance. So the previous data represent the infected versus uninfected one. Here the data is about infected versus BPQ treated samples where the parasite is being eliminated from the host environment. So in order to integrate, so here we try to integrate both RNA-seq as well as phosphoproteomic data in our to identify the final molecules. So we have taken control and test samples followed by phosphoproteomics as well as by RNA-seq. So we are going to look at both host as well as the parasite molecules. Okay, so before proceeding for the making the samples, we want to assure that the parasite or the treatment has been, the treatment is working. For that, we have done a trypan blue based cell death cell counting assay, and what we found is that after 48 hours, there's a increase of cell death. That means we suggest that the cells are, the drug is working. And in order to check for the parasite clearance, we have used one parasite specific protein for Western blot, that is Theileria annulata surface protein. And at 96 hours, there is a clear reduction in the expression of this particular protein. So that's why we consider this 96 hours time point for both our RNA-seq as well as phosphoproteomics. Now coming to the, so we again follow the same pathway or same data analysis pipeline that I've already discussed in the previous experiment. So we have identified around 616 phosphoproteins in the infected samples, and upon BPQ treatment, almost 300 phosphoproteins are losing their phosphosite. And at the same time, for example, parasite, we have identified 31 phosphoprotein that are present in the control cell, and almost maximum of them around 24 25 are losing phosphorylation on parasite clearance. Now coming to the number of phosphosites, so around 1427 phosphosite has been we found in control, and almost 600 of them are losing phosphorylation upon BPQ treatment. The same happening with parasite, where the loss of phosphorylation is quite drastic as compared to the host. So we again check what are the pathway that has been altered due to this change in or due to this clearance of the parasite. So in order to do a GOBP analysis, we have only took the phosphosite that are losing phosphorylation on parasite clearance. That means it is only present in the infected samples, and when the parasite is being removed, they are getting the phosphorylation is being lost. So we again, when you done this GOBP analysis, we this bubble chart represent the top 10 processes, and I again want to highlight that three thing I have highlighted from my uninfected versus infected. Out of this, two are present or present the same process are being overrepresented in the parasite clear cell, that is negative regulation of apoptotic process as well as regulation of CMK and AMPK signaling cascade, which again highlight that these processes are quite important for maintaining this cancerous phenotype. So again, we did a differential analysis whether any differential phosphoproteins are present upon parasite clearance or not. The left side represent the host molecule. So we have identified some 97 phosphosites that are differentially regulated. And in case of parasite, all the 48 phosphosites are as well, they're losing phosphorylation, as well as there is a drastic change in the expression level as well. So some of the key proteins that we have identified from all our data is that RBM15, I'm talking about the host, that is RBM15, USP8, FOSL2 known as FRAT2, HDGF, and BCLF1. And all these protein has been shown in case of cancer to regulate many different processes. Now coming to the parasite protein, we have identified phosphorylation in micronym ROP3 antigen, that is TA08425. Cyclophilins, again, important molecules. Splicing factor, important for transcription and translation. And some of the key glycolysis molecules, which are differentially phosphorylated, or the phosphoproteins are getting dephosphorylation on parasite clearance. Now coming to some individual molecules that we have identified from our data. So here I want to highlight this micronym or ROP3 protein. So this is a graphical representation of a epicomplex and typical epicomplex and parasite. So this is the micronym ROP3 protein, and these proteins are very important for invasion of eukaryotic cells and establish their niche. So we have identified around 7 to 8 phosphorylation site in case of from our phosphorylation data, and so two sites are being reported for the first time by us, and there is one paper by Wins et al. In 2014 we have also find some similar site as compared to their data. So this orange one are unique to our data, and this green are share between our data and others data. So, however, coming to the biological activity or importance of this particular phosphorylation, no documented instance of biological activity has been present, and this site can be further studied to see the biological importance. However, one site, that is phosphorylation 808, which we found as well as by other group, which suggests that this phosphorylation is important to bind the it's bind to the JNK kinases and playing a very important role in maintaining this cancerous phenotype. So again, I am summarizing my data where I've compared my data with the previously published one. So there is only one paper that I've already highlighted. Before that, only one phosphoprotein data is there, that is cell cycle dependent phosphorylation. And when we compared our data with their, however, I want to highlight that they have only focused on the size and protein. So they didn't find any very high amount of host molecules. However, our data is particularly focused on both host as well as parasite. So this upset R plot represent our data compared to them. So we have reported around 589 host protein, while their data only suggests 62. While coming to the parasite protein, they have reported 55 as it is a purified one, and we reported something around 22 that unique to our site. And when it's come to reported host protein versus our protein, almost 56 protein as they've identified 62 are similar between these two studies. And when you come to parasite protein, only 10 proteins are being similar between our data and their data. As we have identified a large amount of phosphoprotein from the phosphoproteomic experiment in three different conditions, now the questions come what to do with this particular data and what else can be done. So one of the most important thing for phosphorylation is the role played by kinases. So kinases are the important molecules which regulate this addition of phosphorylation. And in case of cancer, uh, the role of kinases or targeted therapy has been very vast in nature. And last year only FDA approved around 43 kinases targeting small molecules for clinical trials. And this is one example I have taken where people have tried to target the MAPK pathway, and these are different kinases that are being FDA approved or are in market right now to inhibit different processes or different kinases for therapeutic intervention. So in order to identify some key kinases, as we have identified a large number of phosphoproteins, so our next goal was to identify the kinases that are maybe regulating this phosphorylation both at the level of host as well as at the level of parasite. So let me first start with the parasite kinase prediction. So this is the basic pipeline we have followed here. There are more than 400 or more than 500 kinases has been reported in case of Bos taurus, that is cattle, as well as human Homo sapiens. However, the number of kinases that are present in case or reported in Theileria annulata is very limited. And one study highlighted that only 52 kinases are present, while the Homo sapiens of Bos taurus are more than 500. So 10 times less kinases are there in the database for Theileria. So what we first try to do is, as none of the database for predicting the phosphorylation to predict the kinases are built for Bos taurus or Theileria annulata, we have considered the tool developed for human or Homo sapiens to predict this particular kinases. As this entry of this particular kinases are in the form of human kinase only, so our what we first did is to find the homology of Theileria kinases in case of Homo sapiens. So we first list out all the kinases reported in T annulata, followed by the protein sequence and perform a blast against the human database to identify the kinases. This, then we use this human kinases to predict the phosphorylation using GPS 6.0 web server. And for a particular phosphosite, there might be several kinases that will be predicted. So to analyze our data in an unbiased manner, we have followed one more parameter. So what we particularly done here is, for example, there is a protein X or site X is present, and around 10 kinases are predicted to phosphorylate this particular X protein. So we have given individual kinase score to for this particular site. So we have taken 1 divided by 10. So individual kinase will get a score of 0.1. So that's how we have predicted this kinases for all the Theileria phosphoproteins. And ultimately, we cumulatively added all the score and give one kinase score to a single kinase. Hope it is clear. If it is not clear, you guys can ask me at the end of the presentation. So using this particular cumulative weightage of each kinase, we have predicted the final weightage of each kinase, and this weightage was again taken to represent the kinase. Now coming to the representation of the kinases. So what we found is that so this particular table represent all the top 10 kinases that we predicted with different kinase score that I will explain later if you guys have any doubt later. Okay. Now, this particular kinase tree represent the total kinases that we have identified from our prediction of the parasite protein. And the circle size itself represent what is the weightage of this particular one. So, what we found here from looking at the data is RIOK3 is one of the highest weighted kinases, and all these top 10 are here we have highlighted. So again, what we try to do is whether the prediction system is right or wrong. So for in order to do that, we again did a RNA QRT PCR to at least check the expression of these kinases in the infected cells. Even the RIO is found to be highly having highest weightage and dominant in case of the kinase. We do not found any expression of this particular kinase. However, other kinases that is TA14255 and other kinases that we find from our data are having high expression in the infected samples. And further validation is needed, and what kind of pathway that are under the control of this kinases need to be studied. Now coming to the host kinase prediction. So around 500 kinases are there. So we directly took the Homo sapiens one. No need to go for homology based search. So we have directly used that particular one. Here we have using the standalone version GPS 1.0.1, and we have incorporated all the sequence at one go. And the software is basically going to give us the kinase, and then we calculate the cumulative kinase score exactly the way we have done it for the parasite. So after I identify this, we have identified the weightage of each kinase and represented in the form of kinase map. And what it represents is CMGC group, CMK are the one that are overrepresented from our kinase prediction. So now coming, okay, give me 1 minute. Okay. Okay, so here I have cropped out the CMGC group of kinases. Hope it is clear from the data. So what we found that casein kinase 2, CDK or cyclin dependent kinases are overrepresented from our data. And we look at the literature whether any such kinases or role of these kinases has been uh, identified in case of Theileria or not. So we have found multiple papers which cite that casein kinase 2 is constitutively expressed in case of Theileria infection cells, as well as the role of CDK has been highlighted in different papers. So these are two screenshots of this involvement of this kinases in case of Theileria. So which again highlight that whatever the prediction we have done from the phosphoproteome data by using GPS 6.0 is being validated by previous studies. However, one of the most important thing what we found from our data is the presence of ERK kinases, that is ERK1 and 2. Hope it is visible here from our prediction. But one of the most interesting thing that we found here is there is this one paper which highlight the JNK where they clearly uh, eliminated the role of ERK. For example, I'm going to read it. Parasite infection lead to an upregulation of all the member of June 1st family proteins. And surprisingly, this occurs in the absence of any detectable ERK or P38 or MAPK kinase activity. As we have found that ERK is one of the key kinase that we predicted from our data. So we try to validate whether whatever prediction we found and whether ERK has any role in Theileria infection is not, what we try to study further in our data. So coming to the ERK prediction, so what we found is that first we check which are the protein that are being phosphorylated by ERK. So what we found that many of the transcription factor which are part of the AP1 transcription factor family, for example, June, BCL6, June B, FOSL2, RUNX3, all these transcription factor are being regulated or phosphorylated by ERK as one of the kinases. So we try to further study at wet lab whether this kinases is really regulating the expression of this particular transcription factor or not. So in our further data, I'm going to look at some of the key transcription factor that are under the control of ERK that we predicted from our uh, kinase data. So the first question we ask from our data is, as ERK is one of the important kinases that has been predicted. So we try to see whether ERK expression is there in our data or not. For that, we have done an immunofluorescence assay where we have taken the activated form of MAPK, that is a phospho-MAPK, and we have done an IFA, and we found that the expression of ERK1/2 is very high in case of the infected samples. The second thing what we ask, there are so many different small inhibitor are present which can be targeted to control the expression of this MAPK. So we have taken U0126, which is one of the important inhibitor which inhibit MEK12, which is upstream of ERK. So targeting this MEK12 will lead to a dephosphorylation of phospho-MAPK or not phosphorylation of MAPK and leading to their decreased activity and decreased expression. So we use U0126 as the inhibitor, and we first followed by the preliminary experiment how quick this U0126 is working in our system. So we have taken our control, that is Theileria infected samples, and we treated our cells with U0126 at 20 micromolar for various time point. We collected the sample followed by a Western blot. So this is wild type MAPK and this is the phosphorylated MAPK. And you can see that the phosphorylated MAPK is present in the infected samples. And upon treatment, at the beginning of 30 minutes itself, there is a clear reduction in the MAPK expression in the Theileria infected samples. So from this experiment, I want to highlight that U0126 can be used uh, at this particular concentration to inhibit the MEK12 activity, ultimately lead to ERK12 inhibition. Now, as you have successfully demonstrated that MEK E0126 can be used as an inhibitor, the next question we ask is what will happen to all the transcription factor that we have highlighted from our study upon inhibiting ERK12 activity. So here I'm going to highlight some of the key molecules that we have validated by wet lab. So first starting with FOSL2, which is one of the AP1 transcription factor, and this IFA represent that the expression of FOSL2 is very high in the infected samples. And when you are treating them with U0126, which is targeting ERK12, the expression is drastically reducing. And even upon clearance of the parasite by using BPQ, there is also a reduction of this FOSL2 expression. And this particular graph represent the quantification of this immunofluorescence images. And in all our data, we have taken at least 50 cells, or here around more than 90 cells in each condition we have taken. And when we have done a analysis, we have found a significant reduction of this expression of FOSL2 in the infected samples upon parasite clearance as well as U0126 treatment. Then our data was followed by other parasite, other host molecules. So here we have used C-Jun as well as phospho-C-Jun, serine 73 portion we have found from our phosphoproteome data. And we are finding the same thing upon ERK inhibition. There is a drastic reduction in the C-Jun expression as well as phospho-C-Jun expression. And this graph represents the same thing, that is the quantification data. Then coming to one more transcription factor, which is RUNX3, we also found the same thing, that RUNX3, here you can see a clear localization in the host nucleus. And upon U0126 treatment or BPQ treatment, there is a drastic reduction in the RUNX3 expression and followed by the quantification. And this is one of the Western blot where I want to highlight that RUNX3 is not present in the uninfected sample where you have taken healthy PBMC. However, the expression is quite very high in case of the control or infected samples, and upon BPQ treatment, it is completely getting down regulated. And one more thing about RunX3, that previously people think that it is a tumor suppressor gene. However, recent findings suggest that this might act as a proto-oncogene in later stage of cancerous process. So all these transcription factors starting from fossil2, C-June, June B, the next three are predicted from from the software that they are under the control of ERK. And upon inhibition by U0126 of ERK, there is a drastic changes in the expression level. And that I have already highlighted that we have identified STAT3 at 705 position. And previous data in cancer human study as well as Bosch Toro study suggests that STAT3 is not regulated by ERK. And from our prediction also, we didn't get that STAT3 is being regulated by ERK. So as a positive control, we want to check whether this STAT3, what is happening to STAT3 upon inhibition of ERK. So this is the control samples, and this is the U0126 treated samples. We have done both wild type as well as phosphorylated one. So you can clearly see that there is no such changes in the expression or phosphorylation of STAT3 upon ERK inhibition, which right now act as a positive control for our data. And whatever the previous reported, we have shown that all these transcription factor are under the control of transcription factor can be verified by using this particular data. As we have highlighted that these key transcription factors are playing a significant role are under the control of ERK. Now the next question comes, is there any gene, what kind of gene expression are under the control of this transcription factor? So in order to do that, so this I am going to, so what we have done is there are some very good public databases, for example, ENCODE as well as CHEA, where people have done ChIP-seq experiment to identify association of certain transcription factor with the downstream gene. And this has not been done for Bosch Taurus. However, this has been done for Homo sapiens. So we have taken all the data of Homo sapiens. For example, let me give an example. For example, STAT3, around 13,915 genes are being identified as a downstream or target of STAT3, and CHEA identified 6013. This way, we have downloaded or taken all the data from public databases. Wherever the ENCODE was there, we have taken ENCODE. Where both ENCODE and CHEA were there, we have taken both. And we identified what are the common genes between this particular database. And we have looked for the common one where it is possible, and where it is only one database is there, we have directly taken all the genes from that particular database. Now, as we performed, I have already told you that we have performed an RNA-seq experiment between Theileria infected samples and parasite clearance sample. So we took that parasite clearance sample or differentially regulated gene and compare whether any such genes are being common between this transcription target genes and our data. So what we found that around 1877 genes are common between RNA-seq and the predicted ENCODE data. And this we have done for all the transcription factor. Then we added one more layer, whether the genes are downregulated or not. So we have taken 1.5 fold as a stringent criteria to look for the downregulated genes. And we performed one GOBP analysis and cake analysis. So what we found from this particular data, I have representing only one of the transcription factor here. So what we found from the data is cell cycle regulated process are being downregulated or are under the control of this transcription factor, and upon parasite clearance they are being downregulated and ultimately lead to parasite death or elimination of the parasite. So we have generated this particular string map of the cell cycle regulated gene in case of the Theileria infection, and the color code represent the expression level. So we've generated this data by using cytoscape. As we are targeting around 6 to 7 transcription factors. So we try to do one more analysis whether any gene is common or common target of all these transcription factor. So what we found is only one gene which is under the control or being regulated by all this particular transcription factor is c-MYC. Okay, so here we have identified all the transcription factor. They are regulated. We also highlighted that they are regulated by ERK, And we also highlighted some of the key processes that are under the control of transcription factor. So now the ultimate question comes is what will happen to the Theileria infected cells if you are inhibiting this particular ERK, or ERK is kind of, is it vital, or the activity of ERK can be hijacked by any other kinases. So in order to do that, what we try to do is what will happen to the Theileria infected cells upon ERK inhibition. So here we have performed on Western blot as well as live cell counting, and we have quantified the percentage of death here. So this Western blot represent that here we have taken uninfected samples. This is TN letter minus represent the uninfected sample healthy PBMC, and this TN letter presence represent that we have taken the infected samples, and we have treated our cells with U0126 for various time point, and there is a drastic decrease in the TSP expression due to the cell death. And we have highlighted that pCNA, which is proliferating cell nuclear antigen and important for DNA replication, is being reduced, suggesting that the cells are being arrested. And the total ERK we have represented here, and the phospho ERK, there's a drastic changes in the phospho ERK level upon U0126 treatment, which I have already told you previously. And one of the protein that we have here taken, and ultimately we are going to represent all the protein in our data. So here we have only highlighted FOSL2, and there is a drastic decrease in the FOSL2 level, which suggests that ERK inhibition lead to uh, cell cycle arrest and ultimately lead to cell death, which suggests that ERK is one of the important kinase for Theileria. So coming to the end of my presentation, so this is the graphical summary, which suggests that upon infection, the ERK is phosphorylated, which regulate the AP1 transcription factor family. Ultimately, it leads to nuclear localization, cell survival, and proliferation. And upon inhibition of this particular ERK by U0126, there is a loss of phosphorylation of AP1 transcription factor family, no nuclear localization, and cell ultimately undergoing apoptosis. So end of my slide. Thank you. Any questions? Hello.
Uh, Thank you. So, if anybody have any questions, they can either type it down or you can say it out loud. Meanwhile, I wanted to know that in your experiments, what have you used as your background protein?
Can you repeat the question?
Yeah, I am saying that in your experiment, in the phosphoproteome experiment, so what have you used as your background proteome?
Background for database search, right? The question.
Yes, yes, yes.
Bostorus as well as Theileria annulata. We have kept both the genome, both the protein.
And so, how have you how have you kept in mind that which means you have got the results for the different proteins? So some of them have been of the parasite, and some of them of the host. So
Yes.
how have you prepared the network, means the network analysis? How have you done the network analysis after that?
Actually, we have analyzed the data individually. First, we went for Bostorus, and
Oh,
second, we went for Theileria annulata. So the two data we have generated individually analyzed the data. We have not taken both the database at the single time while analyzing the data.
and there's the phosphoproteomics. Have you validated it using any is Western blot experiments or gel? Have you validated?
Yes, we have done that. So, for example, so first one that I have highlighted previously is the presence of STAT3. So again, you can see that we have done the immunofluorescence assay. From our data, we found that 705 position is present in the infected samples, and it is not present in the uninfected one. And from our prediction also, we have highlighted that there is not going to impact in U0126, and it's the same thing. For example, in case of other particular protein RunX3, we found the same thing, that RunX3 is present in the infected samples, and upon U0126 treatment or BP clearance, there is elimination of the protein that we have highlighted from phosphoproteins as well as Western blot. Here you can look at that, as well as immunofluorescence assay. So we have done all this wet lab verification for around 4 to 5 transcription factor, along with the kinases ERK1 and 2.
Okay, thank you.
Is there any other question regarding the presentation or any data analysis, something? If anyone have any doubt, kindly leave a message so that I can reply.
Uh,
Archisman, is there any other question from the audience?
uh, I I am not seeing anything in the chat, so and I don't think so. So so Debabrata, I think there is no more questions from the audience, as far as I
Okay.
can understand it. And the presentation, your presentation was very good actually, and it was very interesting because you were working on a unique topic on this proteomics, utilizing a phosphoproteomics. I don't know how many people use phosphoproteomics in India, so it's a very unique thing for us also. And if I get any more questions later than that, I will surely pass it on to you. And thanks a lot for attending for coming and delivering your work. So
Yeah.
thank you.
Thank you, Bioanalytics team, for giving me this particular opportunity. And I think thank you for conducting this particular event.
Thanks a lot. Yeah. So uh, so I think we can close our session here today. And thank
Okay.
you everyone for participating. And again, thanks to Debabrata.